All right, so we left off here, I believe, with this idea of mycorrhizal fungi. We've talked a little bit about their biology and how they interact with the plants, specifically the roots. As you're studying the different, especially like the ectomycorrhizal fungi and the vam fungi or the endomycorrhizal fungi, it'd be a good idea to do kind of a compare and contrast as you're studying what's similar about them, what's different about them, to try to get to the details of what makes them unique. And don't forget the terminology that I pointed out that I'd like you to know as well. So they've known about mycorrhizal fungi for a very long time, but it hasn't been until recently that they've really started to better understand how they're interacting with especially forested ecosystems. They're doing really great work with that. The agricultural field experiments are a bit lagging in respect to that, I think. But with the ecosystems, more natural ecosystems like forested ecosystems, they're realizing that you do have these connections underground that can connect different species of plants in that community. And so it's not like all the red pine trees only form mycorrhizal fungi with all the other red pine trees. You don't have that kind of delineation. You have mycorrhizal fungi connecting diverse species. And so then that begs the question, what role do the mycorrhizal fungi have in the succession of that community, the health of that community? And so there's been a lot of interesting work. There's also been a lot of debate. And I just saw a paper published not too long ago where they were, and I'm not surprised, they were being rather dismissive and saying that some of the work was that they didn't have the best experimental design. They may have a point on a couple of the papers, but some of the other papers I thought were really well done, the research that was published. They also are taking issue with some of the common, more popular books that are out there, like The Hidden Life of Plants by Peter Wohlleben. They targeted that author in particular, which I thought was interesting. And I am not surprised because he writes in a way that makes it easy for the layperson to understand the big picture ideas. So he does use anthropomorphic terms. And from my perspective, I'm okay with that because it's helping get this information out to non-botany people and have them better appreciate what's going on. And hopefully that will lead to the desire to help maintain these ecosystems. So lots of interesting debate. And as more data comes in, of course, that's going to shift one way or the other. But basically what they're showing so far, that the good rigorous science, is that you do have these mycorrhizal connections amongst diverse species. And it is definitely, there's really good science out there showing that the mycorrhizal fungi play a pretty major role in influencing the diversity of plants in that area, the successional process in that area, and ecosystem dynamics. And so finally, of course, I've studied mycorrhizal fungi for a very long time, and I've been very passionate, and I have been rather disappointed in how they have not really been paid attention to in terms of this idea that they are influencing diversity and succession. When you take your basic ecology class, at least when I did, mycorrhizal fungi weren't even discussed. That was not even whispered in that class. I don't know if it's like that now or not. Do you learn about mycorrhizal fungi in Intro to Ecology? Does that ring a bell? What about plant ecology? I'm guessing Dr. Robertus.
To some degree, because when we're learning about like lichen and stuff, like you are learning about that as like it's influencing biological processes.
Yeah.
I feel like by extension they kind of do talk about
a little bit about it anyways.
But they're not telling you what it is.
Yeah. There's so much going on underground that microbial ecologists firmly believe is definitely influencing above ground interactions that we can study easily. But it's, you can understand why the underground stuff has been kind of lagging. It's hard to study. It's really challenging. You have to have good technology and resources and money, and it's just challenging. So I did ask you to watch this video, this TED Talk. She was the one who initiated the science. There's a publication in the Science Journal by Dr. Samard, and they nicknamed it the Wood Wide Web, this mycorrhizal network. And so that was kind of the kickoff to really starting to expand this information out in a more visible way. The journals, the Science, that journal, it's called Science. It's a big, big journal with a big impact factor. If you publish in that, that's a huge gold star for you. And so lots of people pay attention to those articles that are published in that particular journal. And so, you know, her work out west, she's in British Columbia, and she's done a lot of great work with postdocs and PhD students and master's students looking at forest dynamics and the mycorrhizal fungi. So with some of the published papers that have been done fairly recently, this is a 2020 publication, and they've been very curious about phosphorus, how phosphorus gets distributed in ecosystems, because they know that mycorrhizal fungi are really instrumental in helping plants get access to phosphorus. Even though there might be, you know, plenty of phosphorus in the soil, often it's in a form that's unavailable to the plant. And we've talked about that, how it can be precipitated out or complexed with other molecules, insoluble, and you need the enzymes and the activities of the fungi, the mycorrhizal fungi, plus other microbes to get that phosphorus solubilized. Well, they know that the mycorrhizal fungi are good at that. They take up the phosphorus into their hyphae and then transport it to their host. And so here we're looking at the idea of trade symbiosis with its host roots. And so they're starting to look at like economic ideas, biological market theory. So they're applying market theory to biological ecosystems and trying to see if that could be a model to help explain what's going on with a very complex ecosystem. The upshot of this research that they published, it was nicely done, and it was showing that basically the fungi are controlling release of phosphorus to the different plants that they're connected to. They don't always give this steady state amount, and so they can hold some back in reserve and then kind of dole it out. And so it's this much more dynamic interaction where the fungi has more control than they thought. You know, initially they were kind of envisioning, you know, that the fungal hyphae were kind of like a highway system and things just kind of flowed. And maybe it was more due to the plant, that the plant was really tightly regulating based on how well they fed the fungus. Because remember, the plant is feeding the fungus carbohydrate. And so they thought initially the plant was more of the driver of this idea. And now they're realizing the fungi has more of a say in it, so to speak, in terms of how they hold on to phosphorus under like boom conditions, and how they will give it up under crashes in different amounts. And so this idea of trying to look at some of the economic theories and thinking about this idea when you have complex organisms involved, how they might be feeding each other, so to speak. So here, this is looking at the host plant idea now, going at from the other perspective. Maybe the host plant is helping to drive what's going on. And so there's basically a lot of people who study this think that it's not one or the other, it's both. You have the host influencing what's going on underground with the mycorrhizal network and the flow of nutrients and chemicals, but you also have the fungi having a say in it. And so the CMN is usually what you see published lately, that the common mycorrhizal networks, that's how they often talk about it. And so they were using radioactive and stable isotopes to look for phosphorus and nitrogen and carbon flow. And they were doing some, and again, the biological market theory has emerged, they're saying. And so this idea that you can apply that economic understanding, looking at how, our work demonstrates how fungal partners are able to retain bargaining power. And so they're realizing, again, yes, the host is influencing to a certain extent, but the fungi have a lot more say in it than initially thought. And, it kind of makes sense that the fungus is, from a diverse, I think you learn in Intro to Ecology that in general, they hypothesize that you want diversity in these different habitats. Greater diversity, generally they say the greater health of that ecosystem. And so it wouldn't make sense for the fungus to just feed like just the red pine trees. And that's all they do. And every other tree in that system is, oh, well, you're out of luck. That doesn't make sense from a diversity is a healthy ecosystem idea. The fact that the fungus can actually give carbon to a tree that's not getting enough and it's, a minor species, it kind of makes sense that you have that kind of dynamic going on to have a stable, diverse ecosystem. So very interesting, exciting work going on, looking at that the fungi do have a heavier role than what they thought. And there was even this really cool study, I talked about this in Plant Phys too, but this is such a cool study where they were looking at how the VAM fungi in this case were helping mineralize the phosphorus. That means making it accessible and soluble for the host plant, but they were doing it with partner bacteria. And so basically what they see, and I'll show you a video, is that as the fungal hyphae are going out into the soil and finding, they did this experiment where they had patches of phosphorus and the hyphae would get to the phosphorus and they would show the bacteria skating along the surface of the hyphae, and that the bacteria were actually helping to solubilize the phosphorus. And the fungus was actually feeding the bacteria to make sure they had enough energy and enough get up and go to get to the patch, help break down the phosphorus so that the mycorrhizal fungi could take it up and give it to the host. And so here you see this kind of three-way idea of partnership with the fungi and the bacteria underground, working together to get that phosphorus good to go. So here is the video. You see the fungal hyphae. Let me go into let's see, I need to do this. So this is real time. They didn't speed it up. But you can see how the bacteria are skating along, so to speak, along the outside of the hyphae. And they do see like this film of liquid that the fungus has on its hyphae and makes it easier for the bacteria to move. And they also see that in that liquid, there's nutrients for the bacteria to sustain themselves so that they can keep going and make it to the patch of phosphorus. Yes. Yeah, you could consider that to be a biofilm on the surface of the hyphae. Yeah. In the sense that it's a very movable biofilm in the sense that the bacteria aren't staying put necessarily, but it is providing that mechanism. So very well done experiment. So again, in science, it's really, you can't really, you can never get bored. There's always something else coming along that is a new discovery. And, differences of opinions for sure. You always have debate. But this study was very well done, showing the idea of this teamwork effort. I'm going to go back to sharing. Yeah, I know that. Okay. Okay. So really interesting studies. It's kind of in its infancy. There needs to be a lot more work on this to really tease it apart. It's very complex. I give a lot of credit to the scientists who do this kind of work because teasing apart all these interactions is quite formidable. So lots of work left to do. But once again, they're showing that, what's happening underground is critical to what we see, what's going on above ground, and that we have to factor that in. So, okay, you can draw your line in your notes, so to speak. This is it for the exam on Thursday. We're not going to cover the other topics on this slide set. So this is, I don't know what slide this is. Let's see, slide 42 in my slide set. I think it's the same for you. Slide 42. Okay, I am going to talk just a little bit and introduce you to the next topic that won't be on the exam, and then we'll spend some time talking about the exam in general. And so for those of you who have had plant phys or currently in plant phys, this is going to look familiar and sound familiar, and hopefully this will make it so that the next exam coming up in this class, you'll be good to go. You've had it twice. So here, we need to talk about this in this class as well, of course, because this is an extremely important interaction between microbes and plants. Something that has been studied. There's been thousands of papers published on this idea. Scientists have been very interested in this process for a very long time and looking at, if we can better understand this, could we get this to happen in more plants than what we currently find, naturally have evolved to do this with their partner microbes. And so this nitrogen fixation by bacteria. And so this is something that only bacteria can do. All right. Fungi cannot do this. Even though I really think mycorrhizal fungi are the bee's knees, they can't do this. Okay. Are we clear on that? Mycorrhizal fungi cannot do this. I see that sometimes talked about in the general, like, layperson literature, and so no, only bacteria can do this. And what we see is that in this case, I'm going to zoom in on specifically when the bacteria hang out with plants. Okay. And so here you're seeing the bacteria that hang out with plants, they actually have this shift in the root morphology. So these nodules that you see, some people, especially the image on the right, you might think, oh my goodness, this root is infected with a pathogen. These look like a bunch of tumors, and you might have sympathy for the plant, but this is actually awesome. This is a great thing to see if you dig up your plant and you see those roots with those nodules like that. That's what you want to see when you have this partnership going on. Maybe on the left, you could see a little bit that there's a pinkish or orangish hue to those nodules. So sometimes you will see that quite readily. That's what the white arrowheads are pointing to. And so we're going to talk about that a little bit. So only prokaryotic organisms are known to do this. Okay, so bacteria, fungi are eukaryotic. They cannot do this. So there's a range of different bacteria. There's some cyanobacteria that can do this. So they are extra special because not only can they do photosynthesis, they can also fix nitrogen. So cyanobacteria are pretty cool when they have that capability. So there's a term that they use. They call them rhizobia bacteria. Those are the ones I'll zoom in on because those are the ones that hang out with roots. So rhizo means root. And so that term is supposed to get across the point that these are the bacteria that partner up with the plant host roots. So only prokaryotic organisms are known to have this enzyme. I would like you to know the name of the enzyme. Okay, you can either say dinitrogenase or nitrogenase. Either one is fine. And this is a very unique and complicated enzyme, and it allows for the triple bond to be broken. So we did look at nitrogen fixation experiments in lab this semester already. But basically what we were hunting for when we provided that nitrogen-free broth that you put the soil into, we were looking for bacteria that had the enzyme that could break that triple bond and in that process, they end up making ammonia. Then that gets taken up and usually gets incorporated into an amino acid quite quickly. So we're going to zoom in on the ones that hang out with the plant host. So the host in the microsymbiont is often how they refer to it. But basically, we're looking at that dynamic. But having said that, I just want to point out that there are free living. So the bottom half of this table is showing you some examples of free living bacteria that can do this. So they don't always have to partner with a plant. They can be free living and just doing it on their own. Okay. And so here you're seeing, some cyanobacteria examples, Anabina, Nostoc, Calothrix, and then other bacteria. You can see that there's a range of bacteria according to their oxygen requirements. Some are aerobic. That means, they're like us. They have to have oxygen to survive. Facultative, they can take it or leave it. Okay, anaerobic, they don't want to be in the presence of oxygen for any significant time, including some photosynthetic ones that are doing photosynthesis without producing any kind of oxygen in the process. So yes, there's all that going on. So they're just hanging out. Primarily, a lot of them are in the soil and they're just kind of doing their thing. And if they are in a situation where they're assessing things around them and go, geez, there's not very much nitrogen, then they'll go ahead and do that reaction. They'll get their nitrogenase enzyme up and running and fix nitrogen. But if there's plenty of nitrogen in the soil, it would be smarter, really, to just use it and not turn on the genes necessary for nitrogen fixation because it's costly. It costs energy, ATP. So it's not like the bacteria are always doing this. It depends on their environment. And really only when they're pushed into it, which means only when there's an absence of nitrogen, would they then turn to nitrogen fixation because it's expensive, energetically expensive. So we'll hold that thought because we'll talk about that more in terms of how they're thinking about trying to get more nitrogen fixation going on, especially in agricultural fields. Oh, and then of course the top half of this table is looking at some ideas of the plant and the bacteria hanging out. And so you can see some of the genus names here. Rhizobium is in it, Azorhizobium, Bradyrhizobium. And so those are the rhizobia bacteria that we talk about. But, all these others too could fall into that category because they hang out with the roots, okay, of the host plant. Sugarcane, you can see that's a big agricultural crop. The legumes like peanuts and beans and so on, there's lots of different bean varieties. And so they need their partners as well for nitrogen fixation. So basically, if you can imagine, let's say it's a pea plant, peas are legumes. Let's say it's a pea plant in the seed is germinating in the soil and it starts to grow. Roots come out, the radicle comes out of the seed and it starts to grow into the soil and you start to get good root formation, seedling gets established. Well, as that's all happening, you have that seedling sending out chemical cues. They have to be able to find each other. The soil environment's pretty big and pretty complex. And so with a new pea plant in the soil, that pea plant's looking for its partner. And in order to do that, you have to have that communication. And of course, this is through chemical communication that we see that going on. And then you have to have the microbes being able to detect that. And the bacteria have to be able to understand that signal, kind of similar to the quorum sensing idea where we were looking at chemical communication. And so the plant roots will excrete out these chemicals pretty much, waving a flag saying, hey, I'm here, why don't you come on over here? And the bacteria can move up that concentration gradient of that chemical. And that's how they find the roots. And then once they get to the roots, they have to tell the plant, okay, I'm here and I'm a friend. I'm not going to attack you. And I can enter into this partnership with you. All right. And this is all done through chemical communication back and forth. And you have this, the fancy way to say that is that the bacteria do a positive chemotaxis to the roots. That means they're going up the concentration gradient of that chemical that's being excreted out by the roots. Depends on the plant. Some like pea plants, their chemical signal is called homoserine. It's a homoserine amino acid molecule. AA is amino acid, but other plants will do other things like flavonoids, which are actually pigments, but in this case, they're using it as a chemical signal. And so it just depends on the plant. And so these conversations are very specific. So the pea plant, for example, when it puts out that homoserine molecule, it's only going to attract the bacteria that can hear that signal. All right. So it's not going to do all the ones that we showed in that table. Not all of them are going to come rushing to the pea plant. Only a very specific partner, bacterial partner for that particular plant. And then once the bacteria get there, in general, we say that they're excreting out these chemicals called nodulation factors. And I do want you to know that term, please. So nodulation factors are the general kind of umbrella term that we give to the bacteria that secrete out their chemicals to say, hey, I'm here and I can fix nitrogen with you if you allow me to partner up with you. And so the plant has very specific receptors on its roots for that signal. And once that nod factor docks into the receptor, then the plant goes, oh, you're here. Okay, let's move on. And you're a friend. Let's get into this relationship. So one of the first things they see happening is the host root starts to morph. It starts to change its shape. It often will start to branch and curl. And to me, when I look at some of the drawings that they've taken from microscopy work, it almost looks like the root is almost like scooping up and hugging the bacteria, like to keep it in place, keep it there, keep it in close contact. I'll show you a picture. These are the non-factors. You don't, in case for those of you who are visually want to think about it that way, you don't need to memorize the chemical structure. I just put that there if you wanted to see it. So basically, they're similar chemicals, but this tan box where they have it, bracketed with the N subscript, that means that can just keep repeating. It depends on the bacteria. And so you can have, long branches of nodulation factors where it's more of a polymer or shorter ones. It just depends on the bacterial species. Okay. So nod factors, they're a class of lipochitin oligosaccharide chemicals, but nod factors, that's what we're going for. All right. So those bacteria will then, after they've established that they're there at the root and that they're friendly, then the root starts to change. And that's what they're trying to show here. And it almost looks like the root is kind of scooping up the bacteria and kind of holding them. You have to think about, if there's water, rainwater, for example, or irrigation water coursing through the soil, that kind of makes sense that they kind of have that hugging up of the bacteria there to keep them in place. Okay, so now they're ready to start entering into that relationship. And I think I'll stop there. We'll get into that relationship on Wednesday, but I wanted to spend a little bit of time talking about the exam, and you can ask any questions that you have. So I'm going to stop share with this. Okay, so we need to go back to the slide set on microbial habitats and populations. So I'm going to share a different way here. Okay, so we've already covered, oh, what happened there? Oh, I need to do this. I don't know why I did that. Okay, we need to get down to habitats. So it looks like slide 25 onward is where the exam material starts. Why is that doing that? Whoops, sorry. Where'd it go? Okay, so microbial habitats. This slide This section of slides is kind of looking at the big picture. Why is it? I shut that. Screen sharing has stopped. Aye, okay. Come on. I don't know why it's doing that. What happened? Where's my? Okay. So this set of slides was kind of the big picture idea of habitats. There's a chapter posted in the readings and videos section that does discuss some of this. So if you want to read something that backs up what we're going through here in the slides, but remember when you're studying, for the most part, the readings are to support what we're covering in lecture. And so the bulk of the points are really coming from what we've gone over together. So the PowerPoint slides. And so don't get lost in that chapter posting that I did on habitats. So for example, if I didn't really specifically say, I want you to know that section in the chapter, which I haven't done, then you don't need to get into it. All right. So I want you to study the right things. We just don't have time to cover everything, but that chapter does cover some of what we are talking plus other stuff. So just be aware that unless I specifically said, you have to know that, which I haven't done yet in those readings. Questions on any of the information here? Anything that was confusing, you need more clarification on? Why is my screen sharing paused? Why is it doing that? So in the video, you won't see this. Why is it doing that? Or in the Zoom recording. You are screen sharing. Okay. Anything in this slide set confusing? Make sure that you do know the terminology of the different mesophile, psychrophile, know what those mean, but you don't have to necessarily memorize exact temperature. It's more of, we're mesophiles, so microbes that hang out with us are mesophiles, but you have, the basic understanding of what is a thermophile versus a psychrophile is also important. No questions on this section?
Cell counts and stuff.
Comparative ideas is more what I'm going for. So like on this slide, you don't need to know, other than like in general, you see that you have a decrease in cell counts as you go deeper, right? But memorizing exact amounts isn't important. And then this idea of a shift, that's a pretty important concept where the archaea start to take over as you go deeper. They're just better adapted. And understanding why they're better adapted, we talked a little bit about that with their cell membrane ideas and the differences of their fatty acids. What was the next topic? Biogeochemistry, right? Let's shift to that. Okay, where did my... I don't see it. What happened? Oh, there it is. Questions on this slide set? This was, again, kind of big picture idea, getting at some of the bigger concepts. You don't have to know extreme details, but I think, this slide 14, where we look at where are the carbon reservoirs, that's an important concept. So knowing the difference, that it's not just even Steven, no matter what part of the ecosystem you're looking at, whether it's aquatic or terrestrial or deep in the ocean, I mean, deep in the earth crust. So that one's a pretty important one. And then, where I have slides, of course, where I've typed up stuff, those are more things to emphasize. This slide 23 is important, okay? Looking at what's going on with the carbon, hydrogen, and oxygen recycling. Specifically, this one's really focusing in on carbon, but you can't really ignore hydrogen and oxygen, they come with it. So that one's an important slide to think about and kind of dissect and work your way through as you're studying that. Nitrogen cycle, again, like slide 47, we didn't get into the details of that. That's more for a general ecology class, but that nitrogen triangle, I think, is a nice summary of what the microbes are really zooming in and doing. So that's more specific to the microbes and their capabilities. Questions on anything else in here? I think we also looked at the Winogradsky idea as part of hey, what is going on? Where is my Winogradsky? Ugh. I think I'm going blind. Winogradsky. So with this one, okay, just weird. All right, so with this one, I'm not going to really ask you so much about how you did it. But just remember that you were adding sulfur. Okay, so that's an important consideration because really one of the main cycles that allows you to see then, of course, is the sulfur cycle. And so don't worry too much about the sulfur cycle drawing here, like slide 10, where it has all that stuff going on. There's really only a couple of things on there that I wanted you to zoom in on, and that's actually discussed further along here with what's going on with the production of hydrogen sulfide gas deep down in your column, and then the impact that that has as it's diffusing. So thinking about kind of a, the different physiologies going on are definitely impacting part of the successional processes going on in here. And actually also where they end up showing up and thriving will vary, and largely to do with not only their oxygen requirements, but also their sensitivity to hydrogen sulfide, because that is a poison. If you get too much of that exposure, you don't survive, including microbes. So you have a lot of dynamic going on with, you have to think about the oxygen requirements and, where you see full oxygen versus anaerobic, and where the facultatives may end up. Questions on the Winogradsky column idea? More the concepts, not who exactly, like, I don't. To me, it's not as important that you go, oh, I know that the green sulfur are here. It's more of why are they there? Like, okay, they're hanging out there because they're right next to the hydrogen sulfide. They like that because they use hydrogen sulfide. So that's more important than you saying, I know green sulfur are there, because you could say, yeah, I know green sulfur are there, but you have no clue why. And I'm going more for the why. Like, why are they there? What's going on? So you don't have to memorize the group names, but knowing what they're doing in the different parts is what I'm going for.
Should we know why they're like the different colors that they are?
No,
Is that too?
nah. And that just has to do with their physiology.
And then their redox gradient, should that just go along with the fact that we know what the reaction is?
The redox gradient, yeah, basically what they're going here is like, because if you're full on aerobic, of course, then you're using oxygen.
Yeah, so anaerobic to aerobic.
Yeah.
basically.
And then, of course, they put the hydrogen sulfide, because you did add in hydrogen sulfur. So you have deep down the anaerobic bacteria producing hydrogen sulfide. And again, they don't care about it. They're just doing that in order to, they're taking sulfate that you provided and running a reaction that gets them ATP. In the process, they happen to produce hydrogen sulfide. They don't care about that. That's waste. And so it gets excreted. But other microbes can then go, thanks, I'll use that for my ATP needs. And that's what you see in the next layer up. But then as you get further up, some of these are more sensitive to hydrogen sulfide. That's why they end up further away, because of their sensitivity, greater sensitivity. We got into biotic relationships as well, just the basic ideas of where did it go? Species interactions, is that what it was? No, we still had to cover the other one. So that was the one when we were looking at lichens, positive versus negative relationships. We looked at lichens as a good example, and then the slime mold. So that slide set, right? That's on there. I don't know why I'm not finding. This, oops. Where did that go now? So some terms and understanding what that means in terms of how they're interacting. Basic. So these are kind of, again, big picture ideas of how organisms might be interacting. And then we zoomed in on lichens as an example. And endocytobionts. We looked at slime molds and myxobacteria. It's an idea of cooperation. Any questions on that slide set? And then we got into the quorum sensing, how are they doing all this? They have to have a way to figure out who's around them so that they can coordinate their behavior and be more successful at a certain behavior if there's a team versus individuals.
Sorry, for the last PowerPoint, what specifically, I guess, do you want us to know about the differences? Or is it more like just categorical? This is this does this.
So being able to provide examples maybe of like, what would be an example of a mutualism? What would be an example of syntrophism, or if I describe something, could you say, oh, that's syntrophism? Or, oh, that's more of a cooperation idea. So I would, if I describe something, I'll try to give you several clues so that you have some context.
Okay, and then, or just like do like characteristics, like is life cycle important? Or is it, yeah, just like the ideas of what the differences are?
Um, I'm not sure what you mean by life cycles, like, I wouldn't
Like-
go into huge detail on something like this.
Like slide 35.
Yeah, so I wouldn't go into huge details on this, but you being able to appreciate that whole thing going on here is the result, well, after you have the spore germinating, it's the result of they have to find each other, come together, and amass together to form that so-called fruiting body. And so just the basics of it. They've gone into great detail on all the different stages and giving names, but we're not gonna do that.
And then like slide 33, like. You expect us to just like.
Yeah.
cover, like,
Basic idea, yeah, basic idea. So, knowing some microbes have that hydrogenosome instead of a mitochondria, and the basics of what that allows them to be able to do. And then the paused again. Wait a minute. Quorum sensing, nanotubes, and extracellular vesicles. So this is how do they actually find each other when they want to coordinate their behavior, and then what could it allow them to do? And so what's the benefit of doing quorum sensing? So I would like you to know the basic idea of quorum sensing. I don't go into as much detail as I do on that topic compared to when I cover it in Gen Micro, but so kind of big picture idea. And then what I'm more interested in this class, that you understand more of the ecology, like, okay, so yeah, they can do this, but what does that help them do? All right, so what's the point? And then the nanotubes as well, that's a different mode, understanding how that's different from quorum sensing and what it in a big picture idea, what a nanotube is. and how that allows for a different sort of communication. And then the extracellular vesicles that they're starting to really hone in on now and realize that there's a lot of bacteria and fungi that do this too. And that it can actually provide another mechanism kind of similar to nanotubes, but these vesicles kind of free float off the cells. They don't connect cells. And then the microbe plant. Questions on anything with quorum sensing nanotubes, vesicles?
We need to. Would you like us to know what interspecies and interspecies?
Yeah, that's an important ecological concept of. Initially, they thought it was all only the same, like only E. coli cells could do this with each other, you know, like, or only Pseudomonas aeruginosa could do this with each other and not Pseudomonas aeruginosa and Pseudomonas fluorescens, you know, so you have this individuals within the same species. Yeah, they can do it, sure. But they can also do interspecies communication as well. So it's more complicated than initially realized. Okay, well, we need to shift over to lab. You can ask me more questions in lab too, as we're hanging out together.